there's a couple of things that you need to understand about next week's quiz and what direction we're taking and why is all this important to you if you're studying biology. The very first thing that we talked about in the PowerPoint was this idea of this difference between the types of bonding. And the types of bonding are very important because they are how molecules form, but also the fact that these different kinds of molecules act differently. You have to understand that if you're going to try to understand the pieces and parts of the molecular biology that allows you to function as an organism. And the three types of bonds that we've talked about were covalent, and covalent having polar or non-polar. And of course the ionic. You must know the difference between these three. You can look at the slideshow. There's only one, or the PowerPoint, there's only really one key slide to the whole thing. If you understood all the discussion, all the examples that I gave you, really you're looking for that slide that has the non-polar molecule that looks something like that, and then the polar molecule that looks something like this, and it has a partial negative and a partial positive, and the ionic that looks like that, where you have a positive and negative. You have that slideshow with the different electronegativities. Now, it's not important that you understand that a particular atom is more electronegative than another for the purposes of this class. What is important is the idea that you grasp the concept that when an atom is more electronegative or stronger, you get this kind of polarity, and that polarity is important for water. That polarity is important for biomolecules, and it's important that you understand this idea of ions. And you'll see why ions are so important when we talk about solubility and osmosis and diffusion. Okay, in this short video, I'm going to keep tested concepts at the top, and I'll keep adding to it as we discuss concepts that are going to be tested. As I said earlier, the covalent polar nonpolar concept will be tested. Ionic bonds will also be tested. Now remember that the entire PowerPoint is important, but it's most of the PowerPoint are just examples or ideas that help support the concepts that we're talking about here and that we focused on in class, and then I told you we're going to be tested during class time when we went over the PowerPoint. Now you have to understand, when you're looking at a covalent bond, you're talking about a polar covalent bond. You're talking about two atoms that share electrons, and you know this if you watch the video. If you're having trouble with this, check out the video where you look at where you start moving and changing electronegativities of atoms, and you'll note that what happens is that these electrons are going around and around these atoms, literally zooming around in an unpredictable way, by the way, and you'll learn more about that in physical science and then again in physics. But in an unpredictable way, these electrons form what we call an electron cloud. And that's the path of the electron that's not really that well understood, but we know the range at which electrons will travel. And when you have an electron that forms a cloud, this is called an electron cloud model, you'll note that what happens is you get one side, the electrons are going to tend to be more on one side than they are on the other. And these are usually the shared electrons. And these shared electrons, because they end up being over here more often, end up giving this side a partial negative charge and this side a partial positive charge, remembering that this means partial, the Greek letter delta. Now, what that in itself, it would be important in chemistry. Is it really that important in biology? Yes, it's very important in biology because it leads to this idea that particles, because they have a positive and a negative side, can align, and that alignment leads to all kinds of consequences. And it's those consequences, it's understanding that this property, this thing called polarity, leads to a bunch of consequences that impact life. That is the study of biology. That's why it's important in biology. Well, that's why this idea is important in biology. And these ideas, this idea of dipole, this is again, as it was tested in class already on your bell work. It's a dipole force, and specifically probably the most important dipole force as far as biology is concerned is this idea of hydrogen bonding. And hydrogen bonding is important for a lot of things. The key thing, though, is that hydrogen bonding is specifically important in biological systems because it's when hydrogen is bonded to, and remember these lines mean bonded, right? Bonded to something, and that's one of those three things. If it's going to be hydrogen bonding, needs to be either fluorine, oxygen, or nitrogen. So if this hydrogen is bonded to one of these three, nitrogen, fluorine, or oxygen, then this hydrogen is going to have one of these, is going to look very much like this part of the cloud here. So it's going to have a partial positive charge. It's because these three, and especially fluorine, but all these three are very electronegative, this bond is very polar, and this one ends up having a fairly strong partial positive charge. When this hydrogen atom comes close to an oxygen atom from either another part of the same molecule or another molecule, it corresponded to something, usually a carbon, you're going to form a special bond between these two. It's a pull. It's not really a bond. We like to call when things are sharing electrons, we call them some kind of covalent bond, right? An ionic bond is when two things are really strongly held together, like in between two ions, a full positive, full negative. But this is a relatively weak force called a hydrogen bond. It's just very important in biology because it happens so often that it actually defines the shape of molecules. And we know today that molecules, the shape of the molecules, especially when it comes to medicines and enzymes, impact the function tremendously. So it's understanding that this hydrogen bonding forms in between molecules and within a molecule to cause the molecule to bend and to attract and to twist, that makes it important that we understand what hydrogen bonding is and where it comes from and be able to identify when it happens. So hydrogen bonding happens between, again, any kind of hydrogen that might be on one molecule and some kind of either oxygen, nitrogen, or fluorine that might be in another molecule. Of course, this needs to be bonded to one of those three as well. Hydrogen needs to be bonded to fluorine, to oxygen, or to nitrogen. If it's bonded to one of those three, right, and it's near the some kind of oxygen that has a partial negative charge, and this has a partial positive charge because it's bonded to one of those three. Remember, it can only be bonded to one. I'm putting three symbols here because you need to understand that it could be any one of these. So if the word is or, not and, right? So one of these three is bonded to this one hydrogen, it becomes partially positive. And then if you have on the other side of over a small space, this oxygen bonded to some other atom, right? It has to have two bonds, but let's say it's bonded to some other atom. It has this partial negative charge here. Then you can get hydrogen bonding going on in between these, right? That's what we just said a moment ago. So that leads to actually to these particles, and let's say these are the particles here. Let's just go ahead and draw them like this, these molecules. If they're positive and negative, then there's going to be attraction between this partial positive and a partial negative on this side. So every time there's an attraction, there's going to be this pull, and they're going to be held together. Because they're held together, so that when fire is added to it, heat is added to it, all right, these molecules will tend to hold together. They're being held together by this force, and so their temperature, their boiling point will actually be higher than another atom or molecule a group of molecules that are not polar, like so. These molecules, being non-polar, are not going to hold together because there's no partial positive or negative as there is in this molecule here, right? So these molecules are positive and negatives. They held each other together, held together by these polar bonds, these hydrogen bonds, and they're being held together, pulled together like little magnets, where these are not held together. So given the same size, this is going to have a higher boiling point than this. That's important because that explains a lot of what's going on with water and why water can have such a high specific heat, why water can absorb so much energy. And later when we're talking about ecology, it'll explain why areas near the ocean, areas near Great Lakes have more stable weather systems, because these areas near these large bodies of water are able to retain heat, so that it's actually warmer in the winter and cooler in the summer, because it's harder to change the temperature of the air overall, not on any particular day. But because of this water's ability to hold together, it doesn't turn into a gas very easily because it has a higher boiling point. All right. So when you're talking about the application of hydrogen bonding, usually we're going to hear a few applications, and the first one, the one everybody always likes to talk about, is this idea of water, because it's the epitome of a polar molecule, and it's going to line up, and like we said in class, and it's going to define a specific shape, and it's going to hold together, and that's why you have surface tensions. How does a water strider, a bug that walks on the surface of the water, how is it able to walk on the surface of water? Is because these molecules are able to hold each other together using this hydrogen bonding that occurs between this oxygen and hydrogen. Now remember, we said if hydrogen, and this is a great example of what we're talking about, hydrogen bonding, if this hydrogen is bonded to an oxygen, which it is, and it's near another oxygen, which also has a partial negative because it's bonded to hydrogen, then there's a hydrogen bond in between these. And so because you have a partial negative here and a partial positive here, and because it's hydrogen, right, you have this relatively weak bonding or attraction that we call hydrogen bonding. Stronger than a lot of dipole-dipole forces, but a special kind of dipole-dipole force that we call hydrogen bonding. Now, so what? It's water. Okay, well, there's a lot of consequences to that. We talked about a couple of them. You should know them. I'm not sure if I'm going to put them on the test or not, but it's certainly something that could come up on your final exam. And it's this idea that This causes water to hold, because water holds together so specifically, and because as it cools down, these bonds, these hydrogen bonds, continue to hold. They end up forming a shape that's less dense as a solid than it is as a liquid, so water floats, and water floating allows organisms to survive the winter on Earth. Otherwise, the water would all freeze from the lakes and the streams, and the oceans would freeze from the bottom up, and you would have no life. Life just wouldn't be possible on the planet. So that's an important, a very important idea. But putting that aside, one other thing about hydrogen bonding is that as we move forward, all the biomolecules that we're going to look at can do hydrogen bonding. And in fact, they end up being twisted and shaped because of those hydrogen bonds. The epitome of this, the one molecule that, again, this happens with all the molecules, but we can look at molecules. The most iconic of these shapes is this double helix, right? And you've looked at that in a classroom activity. We looked at the phosphate backbone, and we've seen what happens. Is that each of these molecules, let me redraw this, because this, you're going to see it again, and it's actually on the practice exam for your final exam. I haven't gotten that to you because we're going to have an in-service on Monday or Tuesday from the testing office, and I want to make sure I get accurate information to you. When you're looking at this, though, at this molecule, let me draw them too. A DNA double helix is actually made of two molecules. One that goes like this. This is going to be very important as we move forward that you understand this. It's made of two molecules, and where these two molecules come in and how they're bonded and how they hold together is very important. And it turns out that the bonding between these two, these rungs between the nucleotides here, there are nucleotides here, you don't know about it yet, but these bonds that hold these two molecules together, these things are made of hydrogen bonding. So hydrogen bonding is actually the force that holds these things together. So you have to understand that when we're talking about hydrogen bonding, it's an important idea. It also explains the shape of DNA, it explains the shape of RNA, it explains the shape of proteins. So understanding hydrogen bonding is incredibly important, and we'll go. I think we're pretty much done with that. If you have problems understanding what hydrogen bonding is, you need to see me, reference the notes, read the book. No matter what you do, you got to understand what hydrogen bonding is before we take this quiz on Friday, and certainly before the test. Now, functional groups, that's really just about memorization and being able to identify functional groups. And let's go on to the next slide, and we'll explain the 6 functional groups that you're going to have to know. So the functional groups we talked about in class, each one is found in specific molecules, and some are found in all molecules, some are only found in certain kinds of molecules. But it's key that you understand these functional groups, the ones that I want you to memorize, the ones I told you to make index cards for, the ones that are in the slideshow, in the PowerPoint, and the ones I'm going to go over now. You need to understand these because they're going to be the landmarks as we move forward, and we start to say, this is how this DNA molecule makes a copy of itself. This is how you make an amino acid. You have to understand what an amino acid looks like. So in order to understand what it looks like, you have to know what are the components of it. What is it? When you're trying to look at a tree, a lot of people would draw like a little brown stick and some little green fuzzy stuff at the top. Unless what a little kid would draw, use a green crayon and draw around the brown stick. But as you're trying to understand a tree, you're going to have to look at the bark. You're going to have to look at the roots. You're going to have to look at the leaves. You have to really take a close look at it and really start to understand how this particular plant does what it does. You have to look closely at its components, and it's the same thing with anything that with all this biological stuff, all this biochemical stuff, you really have to look at the shapes and the landmarks, if you will, of the molecules, so that when you're given an exam at the end of the year, like you're going to face, you're able to say, hey, that's an amino acid. I know that because there's a carboxylic acid group here and there's an amino group here. Or I know that this is a carbohydrate because it looks like this. Or I know that it's that it's a lipid because it looks like this. Or I know that it's a nucleic acid because it looks like this. So you need to be able to understand these functional groups, and you need to be able to identify these molecules. Now, someone mentioned earlier, I think it was Thursday, these functional groups, when you said on the study guide you didn't mention these functional groups. And they were right, I didn't mention the functional groups, but what I did put on the study guide was the carbohydrate, was the DNA, was the nucleic acid, was the lipid, and was the protein. Without understanding the functional groups, you won't understand the macromolecules that we're trying to talk about. So again, you need to memorize the functional groups, and then when you're able to understand and memorize the shapes of the macromolecule itself. Okay, so let's look at these functional groups and then make some decisions, and hopefully try to understand what's going on with these functional groups. One of the first functional groups you learned about was this carbonyl group, and you have carbonyl oxygen. It's a double bonded oxygen to a carbon, and this carbonyl group, this oxygen can be bonded to a carbon that can be bonded to anything. If it's a double bonded oxygen in the middle of the molecule or at the end, but if it's a double bonded oxygen, it's called a carbonyl group. Now, there were two that we discussed. The two subdivisions of this group called carbonyl group. We're not going to worry about those, the ketones, and we're just not going to worry about them. We're going to focus on this idea that if you see a double bond O connected to a carbon that's in some kind of bonded to some other carbons or hydrogens or oxygens, but if you see a double bond oxygen to a carbon then to some other part of the molecule, we're going to call this thing here carbonyl. Now, if that double bonded oxygen is to a carbon, now this is a tricky thing, and I've explained it a couple times, but it's difficult for a lot of students to understand if they forget it often. Whenever you see a diagram, a drawing, where there's sticks connected to each other, we call the junction of the sticks, it's assumed is a carbon. All right, so when you see this here and you see this junction where these two lines come together, there's a carbon there. Hydrogens are never shown on these drawings unless they're connected to some other molecule other than a carbon, or they're in the middle of a special group. This particular hydrogen they chose to show, in this particular drawing they show all the hydrogens. But often when they draw these stick diagrams, the diagrams don't have the hydrogen shown, and nor is the carbon shown. This is an exception here, then this is an exception. The reason they made that exception is so that they can show you something, and we'll talk about it in a minute. So when you see a diagram like this and you see all these junction between the lines, each point is a carbon, and each carbon has four bonds available. So if you see here that this particular carbon, it does not have a hydrogen connected to it. It only has two bonds. It looks like it only has two bonds, but it doesn't have two bonds. We know it has to have four. So this one bond is connected to this other carbon. How do I know it's a carbon? It's not labeled, and there's sticks coming together to it. And it's also connected to an oxygen on this end, right? So where are the other two bonds? Well, the other two bonds are hydrogen. They just assume that there's hydrogen connected to this, taking up the other bonds, if it's not shown. Sort of like the 1 that's in front of the variable in algebra. So in any case, these are carbon carbon compounds. Each junction is a carbon. You can see here, this would be a carbon, this is a carbon here, and this is a carbon here, and this is a carbon here. Any bonds that are less than 3, less than 4, like this one here, or this one here, and it doesn't show what it's bonded to, then you can assume it's a hydrogen. All right, so that being said, let's talk about some of these biomolecules. The first group, as we said, is carbonyl group, right? So we talked about that. The next is this hydroxide group. So anytime you see an OH, there's an OH here, that's a hydroxide group. There's an OH here, hydroxide. Here's an OH here. Let's see, where is there another OH? Here's an OH over here. It's a hydroxide. So a hydroxide is anywhere you see an OH. OHs, hydroxide is you combine a carbonyl group with a hydroxide group, and you get carboxylic acid, or carboxyl. It's not very imaginative, but it works out really well. Carboxylic acid, or carboxyl group, is a combination of this double bond O and OH. So this would be called the carboxyl group. If I put this on the quiz, I would expect that you're not going to label this as a hydroxide group or as a carbonyl group, but rather you're going to look at the entire thing and call it a carboxylic acid or carboxyl group, right? So this being on there, we call it a carboxylic acid. The next functional group I'd like to talk about is this amine group. Here it is. It's just a nitrogen bonded to other structures. Just call it an amine group. Usually it's bonded to this kind of carbon, and then you'll have either 2 hydrogens or 3 hydrogens, depending if it's charged or not. And the name slightly changes if it's charged or not charged. But for the purposes of this class, just call it an amine group if you see this nitrogen bonded to other structures. And remember that R is the variable here. R is a variable. So that brings us to this. This brings us to. Oh, hold on a second. Let me change the. Where do I can't change it. This brings us to this molecule here, and this is an amino acid. And the reason it's called an amino acid is that you see the amine group, that N, see N bonded to other things, whatever they are. Here's N bonded to 2 hydrogens and then the carbon. Now, what you're going to notice a lot of times is that they look different. They're not all going to look the same. Look at all these different ways of drawing it, whether it's a colorful way over here, or just stick with all everything labeled, or maybe there's sometimes not, the carbons are not labeled, as you see here or here. But sometimes you have these beautiful little drawings that show you the double bonds clearly. This carbon clearly shares 2 bonds with oxygen here. When that's drawn, it's drawn for you so that they can teach you something. Usually when scientists are drawing it, they're going to leave out as much as they can, because people know that this is C double bond OH. They might actually just write COOH, or they might just write AA for amino acid. They know, what it looks like. They're not even going to bother with the structure. So when you see this, and also it depends on which side is which, right? So here you see C double bond O OH, and here you see the double bond O OH, but there's no C listed here. It's assumed you're gonna know it's a C. So here you see C double bond O OH, and then it's on top. It's not on the side. It can be in any which way. There's 3 dimensions in space. The real world has 3 dimensions, and these molecules can be in any direction, and the direction in which they're drawn usually means something to where they're going, where they're building the molecule, and what it is that they're focusing on. So when you're looking at this particular molecule called an amino acid, it's made of 2 major functional groups, a carboxylic acid and an amine group, and it's called an amino acid because of that. All right. Now, when you put a bunch of these amino acids, this is a monomer. Amino acids are monomers of something called proteins or polypeptides, and you should know that we will cover it in class. We've hinted at it. You have a video that was emailed to you in Jupiter Grades last week. This asked you to go over this stuff when we talked about hydrolysis and dehydration synthesis. That when we take these monomers, we can put them together one after the other, and we can create a protein chain or a chain of polypeptides by linking them together using that dehydration synthesis or taking water out by joining the two ends. As you'll see here, that's a C double bond O. That OH leaves, and one of the hydrogens leave on the nitrogen side, and they're linked through what's called the peptide bond, and they continue to be linked one amino acid after the other to make a long chain. So that's an amino acid. Remember, the R group is a variable, as it is here, 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 and here. That's why they put these different little tags on it, these little apostrophes, single apostrophe, double apostrophe. The reason they do that is to distinguish this variable from this variable from this variable. All right, but we always use R in the variables when we're talking biochemistry, or you're talking about molecules like these. The other functional group that you need to know and need to be able to recognize is this phosphate group here. Phosphate's incredibly important in DNA. It's in ATP, and you'll see how important ATP is in just, I think, next week, maybe the week after. It's in DNA. There's phosphate in DNA. This is a DNA molecule here. These are the nucleotides that I kind of hinted at earlier in the video. Here's the sugar phosphate backbone. This is there's two molecules. This is one molecule here, and there's another molecule on the other side. So this is one of the molecules of DNA that's bonded here, and this is another one of those molecules, and they're being held together. They're being held together by these hydrogen bonds that we discussed earlier. So you see, there's an oxygen, and there's a hydrogen that's connected to nitrogen, and that's true of all these hydrogen bonds. This molecule is being held to this molecule by these hydrogen bonds. So if this were an exam, and I would give you these next week, let's say, and I asked you, identify and label, or which of these is a phosphate group, I hope you'd be able to circle, all of these as phosphate groups, as a phosphorus atom P bonded 4 oxygens, and you should be able to identify those quickly. You'll see that there's one at this end. You should be able to identify, of course, here or here. Notice that they look different, don't they? But they're all phosphate groups. They look a little different, each of them, depending on what they're bonded to. But they're all phosphate groups. Now, if I were to ask you to identify, hydroxide, well, you'd hope you'd do something like this, well, this would be part of what? The carboxylic acid group, right? This is another amino acid. Obviously, I hope you could tell that. And there's a sulfhydryl group on this amino acid, the SH. It's a different functional group that we've talked about in class. You should be able to identify it, right? Just like the practice problems we had in class. Here's the amine group, but together this is an amino acid, but this is an amine group. This is the other one's a carboxyl group or carboxylic acid. Together they form all 3 of these groups together form an amino acid. Here's another hydroxide over here. Here's a hydroxide here, here, and here, 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 and here, and here. So all those are hydroxides. There's hydroxides here as well, but this is a carboxylic acid group. Remember, we're going to call it a carboxylic acid group. This here would be called, now that while it's connected, we call it a carbonyl group, right? C double bond O. It's not connected to OH. So this is an amine, right? This is another carbonyl. Here's another carbonyl. There's a bunch of R groups in our variables in there. Here again is a carboxylic acid group here, and here's an amine group here. Together, an amino acid, right? altogether, this would be called an amino acid. Okay, so that's really all there is to functional groups. As far as being able to identify them, each has their own properties. Each lead to certain consequences. But for instance, an amine, nitrogen is not going to be found in a carbohydrate, where you're not going to find sulfur in a carbohydrate either. But you're going to find sulfur in an amino acid, and you're going to find sulfur in, or you're going to find nitrogen in an amino acid and in DNA, but you're not going to find it in carbohydrates. So the functional groups have particular uses. They can form, they can do hydrolysis and dehydration synthesis, which allows organisms to build things and break them down. So you really should be able to identify these groups and start to distinguish between the major biomolecules that we'll be building in the short term. Get to know hydrolysis reactions. Get to know dehydration synthesis reactions. They're explained in the video really well. I'm not going to go on any further. This video is probably longer than I wanted it to be anyways. And know everything that's in this video. And when we go through it over the next week, make sure you can identify all the different molecules and know what they make and how they're used in the body. And you're going to do fine on the quiz. All right. Good luck.